Started, and here we are. And the kind of hurrying this morning, so I didn't update this, but the only thing that's changed is we did the in-class assignment on Tuesday. And students either handed that in or sent me an email with pics of that. And we'll review that a little bit today and go forward on talking about really still setting the stage as we are we are definitely leaning forward into chapter 17, which is about cell signaling and the receptor and signal transduction pathways. And we're doing that because it's going to be a lot for us to understand, but also we can use those examples to answer questions from chapter 4. Should be done reading chapter 4 and then going on to chapter 9. And we have a really solid foundation about chapter 9 because chapter 9 is about transcription regulation. And so that's the clay model video work. And so another reason we did the in-class assignment is to be really solid on that, because chapter 9 adds to that. And we go from chapter 9 to chapter 17. And so that's where we're at. And let me just see what my next slide is to say that, again, Riley was here. And we think of Riley, we think of CAR T-cell therapy. We also think of Riley as a class tutor. And super connect with him. I've been communicating with Riley. We did just a super good job. And he's going to be back 3 more times talking about further into that same topic, CAR T-cell therapy. So I think that's really great. It's a really great opportunity for U.S. students to get a really friendly, clever, I think he's really good at explaining stuff, tutor. So connect with Riley. He's he's working with me on this, right? So you can email me also, but it's just great to have somebody like Riley who took this class last year, and so keep that in mind. Okay, so the drawing assignment we did was to follow up and try to underscore this video and then take a couple additional steps in that context, okay? So in that process, I just want to review the quiz we did, and I sent out an answer key, but we just had to be super solid on this because when we were doing our assignment, there were plenty of questions still like what about this, what about that, and confusion. So just to read through these, which cells in your body have the myogenin gene? All of them. It's because of genomic equivalence. All cells have all the genes. And we're just picking muscle because it's a good example, but it could be any cells in your body and any story in your body. All the cells have all the genes. So every cell has the myogenin DNA, and it's not really changing. It gets methylated in some cells, but it's really just hanging out in every one of our cells. But then which one of them have the myogenin RNA? And the answer is only the cells that are differentiating. I counted it wrong if you said myoblast and realized that that was a little bit difficult because I didn't really lay out the timeline really definitely, but the answer to that is differentiating muscle cells. The cells are done being myoblasts and they're becoming muscle cells. And there are experiments that we're doing in the laboratory class to detect the presence of RNAs, because that's what's going on, right? Is you don't really want to study the DNA, you want to study the RNAs and what's changing. And that's true for every cell in our body. The gene expression changes. Brain cells express brain genes. Pancreatic cells express pancreatic genes. Muscle cells express muscle genes. So then, which cells have the MyoD protein? And it's the same answer, because the MyoD protein activates the myogenin gene and causes it to produce the RNA. So if we can detect myogenin RNA, we can detect the MyoD protein. And vice versa. If the MyoD protein is there, it's active, it's activating myogenin. And it's like an assembly line. You have to keep producing these RNAs because they're unstable. So you're just making an assembly line and it's go, go, go to become a muscle. Then definition of genomic equivalence. And then part of the gene is the coding region in. We're really emphasizing that, right? When Riley comes in and talks about the CAR, chimeric antigen receptor, that's a gene that scientists modified using 3 different human genes and taking parts of the coding region for 3 different genes to make one hybrid coding region. that makes a protein that is a human protein material, but it's 3 parts, and that's because they manipulated the coding region. That's what they have to manipulate that. Okay, and then what's the function of all transcription factors? We talk about them a lot, but they just have one function, and that's to regulate the rate of transcription, like that. That's chapter 9. And why do we need to have satellite cells? So the deal there is that when we have muscles, those muscle cells are differentiated and they can't grow very much. They've left the cell cycle. That's chapter 18. We'll learn a lot about that. They've left the cell cycle, permanently stopped dividing, and so we need some backup cells. And these are these satellite stem cells hang around to help the cells, the muscles heal when we get a muscle injury, like I sprained my back maybe yesterday working out some, and maybe some healing needs to be done, like that kind of injury healing, or we're growing as children, or we're working out and building bigger muscles. So we need those backup cells for that reason. So there are some other tissues in our body that can also heal. Sometimes there's different explanations. Our liver can heal. It does a really good job healing from damage. But it doesn't exactly have satellite stem cells. It doesn't have stem cells at all. Then other tissues can't heal at all, like our kidney cannot heal at all, like that. So it's a really critical concept in our body's health. Then what is the output of RNA and its DNA and its RNA? And in intro bio you would write answer proteins, because it's all about proteins. But in fact, we have coding genes and non-coding genes. So the bottom line output of DNA is RNA, and then it's those coding genes that secondarily go on to help provide the template for ribosomes to make the protein. And so RNA is the best answer for that. And then give an example in which the myogenic gene would be methylated, and it's a differentiated cell, and it's anything other than muscle. The muscle cells does not methylate the myogenic gene because it needs to continue to use it. Methylated genes are repressed from expression. So the myogenic gene is present in our brain, but it's never going to use it, so it really typically methylates it, like that, okay? And then what happens if MyoD is mixed expressed in chondrocytes? Chondrocytes are the differentiated cartilage cells from the videos, and they have methylated the myogenic gene, so the MyoD does nothing, like that. And I wanted to show these two because these are almost like smarty pants answers, right? Just nothing, like that. That's 2 points, nothing, like that. And answers on the test can be short like that. If you're confident, you're just like, that's the deal. But really, typically, a student will explain them in more words because that's kind of how we learn them. Like we say, it's just this simple, but then we're going to explain it and put it in context and use examples. So just be comfortable with both of those realities, that sometimes you'll know it well enough and you'll just be like nothing. And other times you'll want to explain and give the backstory. And that's good on our exams, because you're helping me see that I understand you know the material, like that. Then what is it, DNA? And I didn't really put the definition down. I was kind of just giving students credit for that. We have a definition we talked about in class, but it wasn't really part of the videos, like that. And we're really using that definition. It's physical, made of DNA, composed of 2 parts, transcription unit and regulatory region, and it affects the phenotype, like that. And then a couple more things. There's more here on that definition. Then what does MyoD protein bind to? Some students just wrote enhancer, but just about every gene has an enhancer. It does not bind to every gene. You would want to see an enhancer of the myogenic gene. You want to be specific about that. And then this is to understand the labeling that we're using of those terms on the clay model. The clay I did intentionally, that's the DNA. And there are some things that are clay, and those include the promoter, that's DNA, the enhancer, that's DNA, the transcription unit, that's DNA. Those are all composed of DNA. Transcription factors are always protein. They're not composed of DNA, like that. The RNA polymerase is a protein. It's not composed of DNA. The mRNA is RNA. It's not composed of DNA. It comes from DNA, but it's not DNA, like that. So that's the intention of those questions. All emphasizing all this is physical, 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 boom, boom, boom. And then these are universal answers. We use the muscle example, like that. But we talked about how you could just change the labels on those videos to what cells you want to talk about. Do you want to talk about the insulin-producing cells in the pancreas? Just change the labels. And you want to talk about the insulin gene, just change the labels, you know, same clay model. It's just now we're looking at the activation of the insulin gene when the insulin producing cells differentiate. Early in an embryo, the embryo is not producing any insulin. It inherited the gene from insulin, but it's not using it yet because it doesn't have those pancreas producing cells yet. Okay, like that. Okay, so and then a lot of times it's like, well, why not? And the answer is there's a transcription factor that's the big switch, like that, like MyoD or mitogenin are those big transcription factors. So I just wanted to roll through that, and then I just wanted to show an example of the drawing site. We got really busy. I know, and mine was really crowded and busy, but part of it was we were breezing through it because some of it was a review, like that, and we wanted to add a couple things to it by making a big image. And so 3 parts, 3 genes. The MyoD, and they're all in the same nucleus. It's the differentiating muscle cell nucleus, and the MyoD gets activated and it makes MyoD protein, and that binds the enhancer of myogenin, and the myogenin protein is also a transcription factor, and it binds to. And when I asked this in class, like what are the genes over here, students had a hard time coming up with that example, so I just want to emphasize that. So this cell is differentiating, it's become a muscle cell. And I used the muscle partly because most students had learned that, oh, a muscle, what do you mean a muscle? Well, that's a muscle, that's a cell that contracts, like that. And then if you look at it under a microscope, it's got this striated muscle fiber in it, like that. And a lot of students have had some physiology either in college or in high school where they just learn, oh yeah, that's a muscle. And that striated muscle is, it's a huge chunk. Like the meat we eat is a muscle. It's just got huge amounts of protein in it, and it's muscle protein. So that muscle protein is made from muscle genes, and there's a bunch of them. So this is just a quick label of the muscle fiber, and this is just a few of them. Oh, and I kind of accidentally grabbed it wrong. Well, we got actin, myosin, tropomyosin, troponin T, and titin, and so forth. Just lots of different proteins, each made by a gene. So one of the take-home messages from this is that we talked about on our drawing, make lots of molecules of myogenin. There are tens of thousands of molecules of myogenin get made. Myogenin can bind to the enhancer of all those muscle genes. There's always plenty of the transcription factor. We also drew lots of dots for MyoD. Anytime there's a transcription factor, we might just draw one molecule in our model, but in fact there are thousands of them, and they can activate a bunch of genes, like that. And so let me show you an example of that. Let me see how far we have to go to get to it. From the clay model videos, I say, this is a mouse that was genetically modified to have some modified genes that would glow when certain cells differentiated. And the one we're interested in is that the muscle cells glow red. But earlier in the embryo, they have that gene, but the gene doesn't glow. It's the protein product that glows, and it's not being made yet. Because that glowing gene, the transcription unit for that, was actually connected to the myogenin gene. So I just want to draw that and just show you that idea. So here's a mouse, and this mouse has all the genes, and it's a wild type mouse.
Can you define wild type?
Yeah, wild type mouse means the one that you catch in your basement. It just means it's a pretty normal mouse, not a weirdo mouse that has mutations and is studied in a lab. And so wild type is just a generic term from genetics, meaning just a normal mouse with all the regular genes. So of course it has this myogenin gene. And the myogenin gene is composed of the transcription unit that's going to get transcribed during muscle differentiation into myogenin mRNA, and then the ribosome will translate that into myogenin protein during muscle differentiation. And the myogenin gene has the promoter there in the regulatory region, and it has this enhancer, and it's activated because the MyoD protein is bound to it. Okay? And so over here in a test tube, and I described this in the video, but we can understand it more fully. Over here in a test tube, the scientists have that myogenin gene, DNA, and they have it isolated, purified away from the organism, and it's made up of those two parts. And what they do is they take the regulatory region and they throw away the transcription unit, DNA, and they replace it with another transcription unit, and that's this gene that can make a glowing protein. And the gene is called GFP. Normally you learn about it, it glows green, but there are mutations of it that glow different colors. So ours is going to glow red. And so they connect those and they create what the general term for that is, it's called a transgene. And a transgene is a gene that was designed in a test tube. And what they do is they take that DNA and they get it back into a mouse. So there's a little mouse. And that mouse they get it back into is called a transgenic mouse. And it's a normal mouse. We've just added an extra gene. And it's in the chromosome. So this transgenic mouse is called a transgenic strain or line, because we could breed it forever and its offspring will always have the transgene. And it will be normal, totally normal. It just has this extra gene. And so I'll just draw the gene down here, and it's got this extra gene in addition to all its regular genes, including myogenin. And so here's the transgene in the mouse. And because there's plenty of MyoD in this transgenic mouse, when muscles differentiate, MyoD binds to the normal myogenin gene, but there's plenty of it to also bind to our transgene. And as a result, in this mouse, anytime we see myogenin made, the cells also make this GFP protein, and they glow red. So we could use this mouse to study myogenin expression, because anytime myogenin is expressed, the cells glow. Anytime, anywhere, like that. The early embryo of this transgenic mouse, not glowing, but before the heart starts to beat, the cells start to glow red, because they make myogenin and they're activating expression of these of the myogenin gene. And it's both the transgene and the wild type myogenin gene get made. So the mouse is fine, making muscles, doing great. They just glow, just extra glow like that, no big deal, okay? So people call this transgene, they like to call it a reporter gene, because it's going to report to the scientist anytime, anywhere myogenin gets made. So if you're studying myogenin, it's a powerful tool, this transgenic mouse. You could get it, and study it and help. So it helps us study myogenin. It also helps us study the muscle, because it always glows as a muscle is getting made. It also helps us study MyoD, because anytime MyoD gets expressed, the gene is going to activate it. So we can use it to study MyoD. If the cells are not glowing, MyoD is not active. And for us, that MyoD example is pretty simple. It just gets expressed and it's just active. But remember, we've already talked about some transcription factors that get switched on and off, like FOXO. And we could make a transgene like this that could help us study FOXO. Instead of using the myogenin regulatory region, we'd use a gene that has an enhancer that FOXO binds to. And then anytime FOXO is activated, those cells would start to glow. Or we could use it to study insulin, because we could take the insulin transcription unit and replace it with the GFP transcription unit and add that transgene, and anytime insulin gets made, the GFP would also get made. Or hemoglobin, or any gene. We just take the transcription unit for the GFP and hook it up to any gene we want, and we can get those cells to glow anytime that gene would be expressed. It allows us to study that gene. It allows us to study the function of that gene, like making muscles. It also allows us to understand and study the upstream regulation of it, like that, okay? So that's a reporter gene, and the tricks there are built on what we know, which is all genes are made of two parts, and we can mix and match them in test tubes and make transgenes and transgenic organisms. It doesn't have to be a mouse. We can make it a fruit fly or any other species. Always the same concept. Let's just mix and match transcription units and regulatory regions. And we can use this regulatory region of any gene to drive the expression of this glowing gene and study it like that. And so you've heard about glowing genes. You know, you can even buy them at a glowing fish and organism. You can buy them in a pet store. But this is the scientific value of this, and the scientists that developed that logic and strategy won a Nobel Prize like that. You know, because you're just like, cool, powerful, powerful. That reporter gene concept is powerful like that. Yep.
For the MyoD, is it like binding just to one regulatory region and then that's gonna switch on like transcription for both the like wild type gene and the glow, or does it have to have two?
There are two separate genes, and there's plenty of MyoD,
Okay.
right? And now that we have that, MyoD can activate every gene that has an enhancer that MyoD binds to, because there's plenty of MyoD. We can go back to our drawing a little bit, and we can see, myogenin has no problem activating many muscle genes, because they have enhancers to myogenin, and there's lots of myogenin. So it just binds to all those and activates a whole bunch of it. And that's really beautiful, and that's true for a differentiation of any cell type. In muscles, when they differentiate as muscles, we express a whole bunch of new genes. And those new genes would be called muscle genes, because they're made in muscle, and they make the muscle proteins. And it's a bit of a circular definition. What's a muscle? A muscle is expressing muscle genes. What are muscle genes? They're made in muscle. Like that, okay? This is true for every cell type. Pancreas, liver, bone, everything. They have what are called tissue-specific genes. Brain-specific genes, muscle-specific genes, liver-specific genes, like that. So, and it's no problem for these transcription factors to activate all of them, like that. Switch them all on. Our story is still a little bit simplified. There are some additional transcription factors that are helping get the job done. But that's just, more advanced. It's just more add-on. That it's not just myogenin working alone, and it's not just MyoD working alone. There's several transcription factors playing, overlapping roles to get these jobs done. But the emphasis is that there's lots of that transcription factor, so it can activate all the genes that have an enhancer for it, like that. Okay. And so that's that. And all right, and then the other thing that we were talking about is these big arrows that I drew at the end. And so these big arrows are like a pathway of gene expression, and when they're transcription factors, it's a feedback loop where we have the steady, stable, unchanging DNA and these dynamic expression pathways where MyoD DNA gets expressed into a protein onto the cytoplasm. Now it goes back in the nucleus and activates myogenin. Then myogenin gets expressed and made as a protein in the nucleus or in the cytoplasm and comes back in the nucleus. And so we have those as this basic example. And then we have these transcription factors that when they're made in the cytoplasm, they don't just go back in the nucleus right away. Like elk, it needs to be phosphorylated. So this loop can be regulated by regulating the transcription factors, switching it on and off. We'll learn about several other transcription factors on our list, right? We've got about 4 of them on there now. We'll have some more. Estrogen receptor is a protein that's a transcription factor that regulates all of the responses to estrogen. All of them. Same for testosterone. And so estrogen receptor gene, when needed, gets made and translated into a protein, and then it's inactive until the estrogen hormone binds. So that would be another one on our list. It's dynamic because of hormone. And then another one is what we've already seen in elk. We'll see more examples in chapter 17 where signaling molecules, like a hormone or like PDGF, through pathways of signal transduction, activate transcription, more and more transcription factors like elk. So we're going to add to that. And so this is also, like you can zoom in and know all the details, the names of the parts of the DNA for the genes and the enhancers and stuff. And then you can step back, right, and see that this is a general model for pathways of gene expression and how they're dynamic and regulated and for our whole course, okay? So for example, we talked about elk. And so we could have drawn this, and it could be what? It could be skin cells under a wound, like that. And this is the normal skin cell, and then the wounded skin cell. And it's got to stimulate the cell cycle, chapter 18 topic, by having this PDGF regulate ALK. And then ALK is going to regulate, in this case, not muscle differentiation. ALK regulates the cell cycle. It gets the skin cells to grow and divide. Like that. So it's the same drawing, same image over and over and over and over again. Thousands of different genes and different transcription factor stories plugged into that same broad image, like that, okay? All right, and then just to show you this, that when we talk and zoom in on a gene and talk about transcription regulation, that's chapter 9. And then when we say, oh, there's a transcription factor regulated by some signaling molecule, that's chapter 17. And then if the cell response in this target cell is to go through the cell cycle, that's chapter 18. So we're putting together a bunch of chapters in this one image, and that's why we started our course. I started our course and I said, there's a lot of background information, because this is really intro bio topics, but we stack them all together. Intro bio is an introduction to this course. It's weird. But it's true, every college and university in the United States starts with a first semester of intro bio for majors that's an intro to this course. It's entirely focused on this course, like that, right? And it's because in the last 50 years, billions of dollars have been spent by the federal government on this war on cancer, and huge amounts of money have been paid to these scientists that are just pushing this topic forward. And it's not limited to cancer, because these are just universal diagrams of how gene expression works in all of the cells in our body and every cell on Earth, like that. So it's just a spectacular situation, okay? So then, just to remind you, we have some terms that we picked up, like cis and trans, genetic terms like that, and the idea of the terminology for these things. And I said that the terminology for the DNA parts of a gene vary. They vary from biological topic to topic, and that's a little bit unfortunate, but this is a course where we can recognize that they vary a little bit, and we'll use them and we'll talk about them a little bit and make sure you don't get confused. We have a nice set of core terms: transcription unit, regulatory region, promoter, enhancer, that we can use consistently. And every once in a while we'll say, in some areas of biology, the scientists really just call this a little different, use a little bit of a different term. And our author does that some, and doesn't apologize for it, but it can be frustrating like that, okay? So that's where we're at. And so let's just pause here after that assignment and this review of the assignment, the goals for that assignment, and see if people have questions about that stuff or anything else at all where we're at. Yep? This one or earlier?
No, further back. Sorry.
No problem. That's probably the first image. I was just putting up the kind of the homework assignment.
Okay. It was one that you had drawn, like the first drawing for the day with the GFP.
Oh, maybe over here?
Yeah.
Yeah, sorry.
Do you mind just describing the parts of the last
The transgene? Yeah, so I'm going to erase it, but we're going to go back to this, or maybe I don't need to erase it. So we're going to talk about the transgene. And the transgene was prepared in a test tube, and the scientists call it gene splicing. Or they call it recombinant DNA technology, because they're rearranging genes. That's what Riley's talking about with CAR T-cell therapy. In that case, rearranging the coding region. In this case, taking advantage of the fact that a gene is composed of two parts. The transcription unit, the business part, the output part, the functional part, and the regulatory region. And what we're doing here is, in the test tube, is we're taking the regulatory region from one gene, in this case myogenin, and we're just connecting it to a transcription unit that produces a different product, in this case, produces a protein that will glow. And so the names would be the same terminology. So this would be the GFP transcription unit. And so within it, that's the transcription unit. That's part. It's going to get transcribed by RNA polymerase and make mRNA. Within it, there'd be a coding region that's got the coding information the ribosome will use to synthesize the GFP protein that will glow. And then next to it, we have the regulatory region that was really from the myogenin gene. It's got a promoter that transcription factors are going to bind to show the RNA polymerase where to start. And then up here, it's especially important that it's got the enhancer that MyoD will bind to. And because of that, we cause this transgene to be under the control of MyoD. So where MyoD is active, we will see the cell glow. And before it's active, it won't glow. So we could do a test and we could say, are those cells in an embryo glowing yet? No. Do they have MyoD protein? No. MyoD RNA? No. MyoD gene DNA? Yes, they always have that, but they just haven't started expressing yet. Then, once the cells start to glow, do those glowing cells have MyoD? Oh yeah, because it's just a real cause and effect, common sense matter of fact. MyoD is going to bind to that enhancer, that transgene, and activate it. And we'll be able to detect the glowing protein, we'll be able to detect the RNA for that gene, like that. Yep. Yeah, and don't hesitate to ask questions. Because this is, like I said, it's just crazy how this is positioned in the field of biology, right? Every college and university teaches this first. And it's such an intro level that you don't really get what's going on. But it's these people that are just like, what do you think we're up to? And you're just like, whoa, you guys are busy. Yeah, because it's thousands of scientists, with billions of dollars of money figuring this stuff out, like for cancer and then everything, right? And that's, scientific discovery is its pace of it is based on how much money is focused on it, right? So the science was pretty slow until the 1970s, and Richard Nixon, that's the president that declared a war on cancer, like that, and just started pouring money into this, and it just continued like that. So it's a lot of money, like that. Just that's the situation. Yeah. So other questions about any of this stuff at all, and anything else. But you'll see we're just going to casually refer to this stuff as we're going forward. Be like, oh yeah, we're talking about. And when we do, you can back up and you can say, hold on, I want to check in and ask about that. Let me give you an example of that. Okay, here's an example of that. So I'm going to scroll forward a ways because I've just came up now that it's a good time for us to deal with this. Scroll forward to this. So here's that pathway, the normal healing pathway found under a scab. And we've got our terms. We've got PDGF, platelet-derived growth factor, PDGF receptor, that's going to come up in chapter 17. It's a receptor, tyrosine kinase. That's the type of receptor the receptor is. And then it's an example of a signal transduction pathway where we have famous RAS and I put MEK kinase. Don't just discard that. RAS, RAF, MEK, ERK, L, like that. Okay, that's normal. That's healthy. Okay, right? Okay. So here's a healthy person like us, right? In that skin cell, there are all the genes. There's the PDGF gene. It's not being made. The PDGF gene gets made in the bone marrow by the cell that makes platelets. That's where we get the PDGF. It's in the platelets, and at the site of the wound, the platelet releases clotting factors and PDGF. the skin cell has it, okay? All right? Here's a woman that has a very, and you don't need to write this name down, but she has a very, very rare specific type of skin cancer. And doctors recognize everybody that has that very rare kind of skin cancer because they'll do a biopsy, they'll take a little bit of the cells, and they'll look under a microscope. And they can actually see everybody that has that particular type of skin cancer has the same problem with their chromosomes. You can see it under a microscope. So this is a karyotype looking at the chromosomes. And here's what happened in that woman that is causing the cancer. So she's got a mutation. And you learned in intro bio, there's some different kinds of mutations. There are point mutations that can change the DNA and the coding region and change the amino acids. We talked about those kind of mutations for RAS. The RAS protein is an enzyme that turns itself off. And there are point mutations in the DNA, in the coding region, in that catalytic region, that catalytic domain that's used to turn itself off. So they inactivate the turn off. That's where those are. They're in the coding region, it's a defective protein. This woman has a different kind of mutation, and it'll be one where we'll just sigh and we'll be like, oh my gosh, when we learn about it. It's a different kind of mutation. Here's what happened to her. Instead of point mutations, what happened is the DNA got torn. So the DNA is double-stranded, and there can be mutations where there is covalent bond damage, like that mutation, that breaks a strand of the DNA. That's called a single-strand break, or a mutation that can break both strands. That's called a double-stranded break. It's a different kind of mutation, and normally our cells repair the damage. And the way they do it is they get to both ends and they glue them back together. Unfortunately, in this woman, she had 2 chromosomes break at the same time, just randomly, and when her body fixed them, she glued the chromosomes back together wrong. And you can see that under this microscope, because you can see the chromosomes and you can order them and label them, and you can see that there's been what's called a chromosomal translocation. That part of one chromosome got joined to another by accident. It was an attempt to repair the double-stranded break damage of 2 chromosomes, but she did it wrong. It's just like if you took your biology book and just accidentally fell down the stairs and tore it in half, well, you'd glue it back together. But what if you fell down and you tore your biology book and your math book? Then you glue them back together and you might get them wrong. And you just be like, well, there's a weird textbook. Look, we're talking about this, and then it just jumps into math. I don't know, right? That's exactly what happened to her. It was an accident. It was an accident like that. Random error, okay? But here's what happened. The part of the one chromosome that got torn is the transcription unit for PDGF, and the other chromosome that got torn has the regulatory region for a gene called skin collagen. And that gets made a lot in normal skin. And when she glued them together, she made this accidental hybrid gene where she's now going to express PDGF in the skin. And as a result, that one skin cell makes PDGF, responds to it, thinking it's at the site of a wound and it needs to hurry up and divide and heal. And that never stops. So she's got just a broken gene, and it's just never going to stop. And just that one cell has a problem. You know that when cancer gets bad, it can metastasize, it can spread. If this cell moved to the lung, it would just keep growing. It's just a terrible, tragic cancer. And at a molecular level, cancer is always like that. Where you're like, oh my gosh, what are the chances that you would tear the gene, the chromosome apart right there? And the answer is it's very, very rare like that. But that's what happened in her case, right? So it's not a hybrid transgene, it's a natural situation, but it's a mutation in her body. And you can see it. So even back in the 1950s, scientists, doctors could see that, and they would categorize that patient by that way. They'd say, she's got a chromosomal translocation between chromosomes 17 and 22, And we're going to call that skin cancer this long word that I don't know how to pronounce, like that, like that. And that's the problem. And that's the kind of stuff we're heading for. In the, the later chapters there we're getting to, we're going to talk about probably four examples of really famous chromosomal translocations that cause cancer. Yep.
I just have a quick question. What happens with PDGF and people with like hemophilia, where they like can't clot their blood?
So remember that PDGF is a growth factor, not a clotting factor, right? So they have a problem with just the first step of stopping that blood flow and making a scab, like that. And the platelet, clotting factors are proteins, and so the platelet is chock full of a variety of proteins, some clotting factors, and then PDGF and some other regulatory proteins like that. Yep, okay? And so we could draw that out, or you could draw it out on a test. You could say, remember that woman who had skin cancer? What's her problem? And her problem was, she accidentally tore apart two genes, and she actually glued them together wrong, making a translocation. And what she did is she glued the transcription unit of PDGF with the regulatory region of skin collagen. Skin collagen is made a lot in the skin, and so suddenly she's making PDGF in the skin, and it's like, oh, whoops, that's really sad, oops, like that, right?
Do translocations like that happen? Like, it's pretty rare, you said?
Yeah, cancer is super rare, but we have a lot of cells, we have a DNA, we live a long time, and so just random bad things can happen. Another thing that can happen is there are lots of checks and balances in our growth control. So sometimes there's a mutation like that causes trouble, but it actually doesn't directly cause cancer. This sounds like it would just cause cancer. It's just going to be out of control pretty definitely like that, right? Some of the other ones might be ones that run in families, right? So like just about everybody has a family where they're like, well, there's a higher frequency of breast cancer in our family or certain type of skin cancer. Either you know that in your family or you have friends who had to be careful with their son because there's skin cancer that runs in their family like that. And what's happening there is a mutation like this that just doesn't cause cancer on its own, but it sets it up to be more likely to cause trouble because it's made like one of the steps necessary to become cancerous. Like that, okay? So that's just to jump ahead and say, right, based on this, really clear story that we know a lot about, wow, there's this really, tragic translocation, like that. And that's one of them, okay? Okay? I'll give you another one like that as well. So let's go back here. What we're talking about, we're doing great. So I can find where we're at. Okay, so we talked about this and la la la. Okay, so just glance at this, and we'll talk about it more. But I said these are probably the 5 of these 1970s retroviral oncogenes that we'll talk about. One of them is RAS, and we talked about it already. And the key concept is that we will call these oncogenic retroviruses. So cancer-causing retroviruses. They directly cause cancer because they picked up a host gene that is a normal growth control gene, but now it's growing out of control. Like, for example, RAS. One of those oncogenic retroviruses had RAS, and it was mutated. So it was that defective RAS that couldn't turn itself off. We'll talk about another one in chapter 18 and 20, and it is MYC. Okay? So MYC codes for a transcription factor, and it is activated when the cells are stimulated to grow and divide. Okay? MYC was originally discovered as in an oncogenic retrovirus in the 1970s. Okay? Here's the deal with MYC. MYC isn't mutated in the oncogenic retroviruses. It's normal MYC. There's the transcription unit for MYC, and it's normal regular, wild type, non-mutated MYC. But the regulatory regions for these viral genes are really strong and stimulate high levels of expression. And so we made another hybrid gene with this MYC in this oncogenic retrovirus, where we just took regular MYC and accidentally joined it to the virus, and now the virus is going to force a lot of MYC expression. And that just causes cancer, because MYC is a really tightly controlled transcription factor, and normally there's just tiny amounts, and then it gets got rid of. But for MYC, we made a hybrid gene here, and it got out of control. Yep.
Is that the same myth that activate and phosphorylate ERK?
No. That's RAF MEK, MEK. So this is a totally different protein. MYC is a very famous oncogene. We'll look at it again when we get into a couple more chapters. But MYC is another example of an oncogene that became famous partly in the 1970s from these oncogenic retroviruses, but then especially in the 1980s, because it's one of those examples of a chromosomal translocation. And we'll see that just about everybody that has a certain type of lymphoma has a translocation of MYC, where the MYC transcription unit actually got translocated to have a regulatory region that gets expressed at really high levels in blood cells, and then it's out of control like that. So, that concept of the gene is composed of two parts comes up a lot. Either where we're building a gene in a test tube to make a transgene that's cool to study stuff because it glows, or because it's a kind of chromosomal translocation. And we'll look at 4 stories of those, I believe. And there are several. There are, a couple dozen famous stories of specific kinds of cancers that have this translocation quality to them, that's the problem, is that they got a gene out of control. Okay? So, questions about that? Your question about anything? Okay, so again, we talked about those oncogenic retroviruses. That's what we're going to call them, right? Retroviruses are a certain category of virus, and of those, some of those retroviruses, not human, were found in the 1970s to have the ability to just cause cancer immediately upon infection. And then in the 1970s, scientists like our young author found in Wisconsin that it was a host gene that was causing the trouble. And then it's really important that we understand then we leave that story behind. And the author did by going from Wisconsin in his graduate work to Boston, because he left the viruses behind. And what he did is he took mice with cancer, purified their DNA, and injected parts of the DNA into healthy mice. And he found that that DNA could cause cancer. And when he looked, he identified that RAS gene. Nothing to do with a virus. The viruses were a 1970s foot in the door. They found some really famous ones like MYC and RAS and so forth. They move on, and it's just now cancer. And these are genes that are often mutated in cancer. RAS is mutated in a third of all human cancers. Nothing to do with virus. Just random mutations, point mutations like you learned about in IntroBio, in the coding region of RAS that make it defective so it can't turn itself off. Okay, so that's our author in Wisconsin studying oncogenic retroviruses, getting a handle on RAS, going to Boston, and learning about that. So we learned about the replication of that retrovirus, and the reason we learned about it is because of the reverse transcriptase that's used to copy the virus in the replication cycle, and then this key concept of that the retrovirus gets incorporated into the host chromosome. So that's where it bumps up against host genes like RAS or MYC, and where we could make a mistake and take that host gene with it to the next cell and infect that cell with this dangerous gene. Okay? So just as a quick question, clarification, here's the replication cycle for COVID. It's not part of our course, we're just glancing at it. And it just involves infection, and it's a DNA a virus, I think, or whatever, and it just gets copied and it goes on. It doesn't get integrated into the chromosome. Just based on that brief explanation, do you think COVID could somehow get in a situation to cause cancer like these oncogenic retroviruses? Why not?
Because it's never inserting into the genome.
Yeah, the COVID virus replication never integrates it into the genome, so you'd never have that accident happen where you're like, oops, it just couldn't do it like that. You know, and it just couldn't do it like that. So the retrovirus has in it, and this, our author's boss in Wisconsin, he won a Nobel Prize because of the pathway, and the pathway is that reverse transcriptase, which is fascinating. But then you see how it's getting into the DNA, so it could pick up a host gene, right? It has to have that step in its replication cycle to have this problem come up. Very rarely, accidentally, oops, picked up the RAS gene, picked up MYC, yeah.
So is the reason why, like, a retrovirus can insert into the genome and that, like, COVID can't because retrovirus codes for the reverse transcriptase and COVID doesn't?
It's the second reason, really, is that when the retrovirus is inserting the chromosome, it takes advantage of the host to make lots of RNA. Mm-hmm. And COVID doesn't want to make a lot of RNA. And I don't know the details of COVID's story, but it doesn't need to do that. And so the retroviruses evolved just, first they got to come in, they got to convert to DNA, then they got to make a bunch of RNA. So how are they going to do that? And the answer is, let's take advantage of the host machinery, get integrated in the DNA, and just be like, hey, express this, like that, for the RNA polymerase, like that. Yep. Other questions people have? Okay, so then there are these different categories of viruses, and we don't need to worry about it, just that we're talking about retroviruses right now. And just for the oncogenic retroviruses in the 1970s, that identified about 30 really famous oncogenes that scientists left the viruses behind and studied in Boston and New York and, Frankfurt, Germany, like that, all over the world, right? So we're just getting started, but we need to understand that, appreciate that. We'll call them oncogenic retroviruses because our author refers to them, throughout our book. There'll be other ones that they mention, and we're only going to talk about those 5, but the author mentioned several of them, and he has a table of them in chapter 20, and, he knows all about them, right? He's been studying those things all his life, okay? So that's our term, oncogenic retrovirus, and should make you immediately think of this figure, and you got that regular retrovirus up there, and then the oncogenic one, because it picked up somehow a host gene, like that, okay? Awesome. And we'll deal with these one at a time. I just like to show that kind of up front, that there's a list of about 30, and we'll learn about 5, and 5 really satisfying ones, and they'll be in chapter 17, some, and chapter 20, some, like that. So we got a ways to go on some of that's after our first exam. So then, this is where I jumped ahead and I said, we're solid in that normal wound healing, and it's an example of the receptor tyrosine kinase pathway, and we've got different receptor tyrosine kinases, and they have different signal transduction pathways with different cellular responses. But that's our favorite one that we refer to a bunch, and we can do it by giving examples like, I guess that picture I might have skipped over it, but the one with skin cancer. We can take our solid understanding of that pathway and say, ooh, look at this situation with her. And we can use that to illustrate the chromosomal translocation, and then this genes have 2 parts. And she accidentally glued together the PDGF transcription unit with the skin collagen regulatory region, and she's going to make a bunch of PDGF in her skin. And that's just a big mistake because she's going to respond. She's got the PDGF receptor, and as soon as she makes it, she's going to respond like that. Okay? Okay, so I just want to show you that in our authors, he studied this pathway that is the wound healing pathway. It's not just the wound healing pathway because there are different receptor tyrosine kinases that use this RAS pathway. So different parts of the body, different normal stories, not just skin. RAS is mutated in 1/3 of all human cancers. So lots of different cells throughout our body use this pathway. using different receptors with different signaling molecules to respond and do different growth things like that. Then also these receptor tyrosine kinases can activate other pathways. Even with the PDGF receptor I just showed briefly, the PDGF receptor is activating multiple pathways. But this is just a sweet, famous pathway like that, okay? Okay, and just to show you, here's this RAS, GTPase, you know, it's a Wikipedia page like that. RAS is super famous, you know? It's just been studied. There's a list of like the most studied proteins of all time. And RAS would be in the top 20. And several of the other ones that are in the top 20 are these oncogenes that are just like, yeah, you know, if you can think of something to study about RAS, we'll give you money because we're worried about it, like that. Yep. Okay, and so our author's that. Okay, so then I wanted to show you this because this is part of what we're talking about, and it's also the big kind of part of take-home message from chapter 4, which, you know, I'm not really talking about much directly unless you have questions. Joe's been asking me a bunch of questions at the end of lab class, you know, and so bring those to class, or we can ask them in the lab, or we can email and meet in person, you know, like that, you know, because I'm like kind of, you know, pretty much done talking about chapter 4, even though there's lots of important stuff in it, like that. Okay, so here's just some key, like, what's the structure and purpose of chapter 4? And one of them is, oh, this is physical. It is so physical, all this stuff. And, you know, it's way down at that molecular level, too small to see, but it's just like that, right? The MyoD has to bind to the myogenic DNA. It has to bind to it physically like that. And so that's one of the big stories of molecular biology, is it's just like car mechanics, nuts and bolts. You're like, oh, you got a problem with your carburetor, okay. Then it's this reductionism where they just go down, just take it down to the simplest level. Instead of starting our course with car mechanics, we start looking at small engines, like a motor on a lawnmower. We say we're going to just take it down to these simple levels. And so those model organisms are a big deal, like down to a simple level. You know, you can get cancer money to study bacteria because of the mechanisms that you're studying, or even the viruses that infect bacteria. Just take it down to the minimal level and really figure out the nuts and bolts, the physical nature of it in great detail. That really emphasizes model organisms. The first chapter says, oh, these model organisms are great. Why? Because of orthologs. Yeah, fruit flies grow fast and they're easy to maintain, but orthologs, they're super closely related to us. We can find orthologs we can find myogenic ortholog. Boom, there it is, right? Easy peasy, like that. Research is done in labs, you know, just controlled environments, model organisms, and breaking them down, you know, nuts and bolts. And then this other thing that the discoveries get turned into tools. That's just a really common thing in molecular biology where they're like, oh, that's cool. Like that GFP. That GFP actually came from a jellyfish that lives in Puget Sound by Seattle. And a marine biologist studied that. He's like, wait a minute, this thing's glowing, and figured out how it glows. And then a scientist at MIT realized, oh man, I could take that GFP gene from the jellyfish and insert it into my little nematode worm and get different cells to glow at different times. And those guys won Nobel Prizes because they're just like, whoa, good job. It's a tool. And so that neat trick thing I want to just show a couple times. So one is GFP. It's just a neat trick, neat, clever trick. The guy that won the Nobel Prize called me at home to talk about that. And I was like, why are you calling me at home? And he hung up. Yeah, I could tell you about that sometime. Yeah, it was pretty crazy. That's him right there. There's the MIT guy. There's a guy from, you know, Puget Sound, from Japan, actually scientist that, you know, got jellyfish in Puget Sound. There's another scientist that won the Nobel Prize because he got the mutations to glow the different colors, like that, okay? And so it's a neat trick. There's the jellyfish, and you know, the details of the Japanese scientists learning about how it glowed and that it was a protein glowing, and then these other people saying, oh, well, we could just take the transcription unit of that DNA. DNA is universal, glue it up to whatever regulatory neuron, put it in our organism, and we can get controlled glow of anything we want. Okay, we should stop and have a little concept chat action. I handed out the sign-up sheet, so find that around somewhere. It's back there. And, you know, try to talk to somebody new, say hi, and say, hey, you know, we should go and we should call up Riley and see what Riley's doing. What's that Riley doing? And we'll just do this for a couple of minutes, and it's, what are 3 important things we talked about today? And if there's an even number of people, and it looks like there are, we'll just have pairs and groups. That'll be great. Thank you. Now the sign-up sheet is up here if you need it.
Yeah, one thing that I read interesting was when he was talking about like oncogenic virus MSC and how it's like he was saying it's causes translocation. Yeah. This is what I
Well, so translocation said it's just part of the DNA for to another gene.
You want to join our? Yeah.
You have similar molecules that like it is like rare and obviously what express them. I was curious and how that can manipulate stuff.
I don't know.
The genetic that's a lot of what viruses do. Like a little bit, and then it's just something you can
I was just curious because I know that he was saying that like
too bad.
Based on like where you inserted that gene, like you can specific.
The output of what the gene is manipulated is not changed too much. And that was just not going to express it at all.
If I understand it correctly, like usually cancer notices, but I don't know if that's an all. Sometimes. Related to cancer. Pretty rare.
Yeah.
And that's similar.
Did you ever share it for your lab? Have you ever done your presentation? Nice. Yeah. No, I haven't.
I've never had.
I think so. It's fun.
And we'll just do this for another minute, and then we'll check in.
Yeah, no, it's cool. You have one day to do it. Yeah. It's tough
And we can check in with Amal. Hi, Amal. What is one thing that you guys talked about that you thought was important today? Yep. So this idea of this transgene, okay, and a transgene is a gene that's been manipulated in a test tube. So we can get the purified DNA out, and we can build whatever kind of gene we want. And those are, the general term for that is a transgene. And then we want to put it back into an organism like that. And that organism becomes a transgenic organism. And there are a lot of different things that we can do with that strategy, and we'll talk about them. The first one we talked about is just, let's make a transgenic organism that will glow, like that will have red muscles that, glow red, like that. Yep. And, that's just showing, and the emphasis of, the DNA is like clay, and we can manipulate it in the test tube, and mix and match, and we have the control of that. And then that's a transgene, and we want to make sure we understand that that's not the problem with the woman with cancer. She doesn't have a transgene. She's got her natural genes, but by accident, she did one of those gene splicing events where she joined the regulatory region of one gene with the transcription unit of another.
So
Yep.
translocation isn't a form of the transgene, so it's not a form, it's like kind of
Correct.
separate.
Yeah. So that's important to understand. Transgenes are built in a test tube. The woman had nothing like that, not a transgene. She's not transgenic. She just has a mutation. The mutation had a similar effect by
Yeah, but
chance.
is it the same kind of outcome where it's like?
Yeah, same logic is we're going to just mix and match the regulatory region and the transcript unit. And in her case, that's a very big problem like that. Yep. And Max had a question too.
Yeah, is there any cases where there's a translocation that happens and then the gene just doesn't get expressed anymore? Or is it always expressed?
Sure, yeah, and you can think about it with regard to the woman because her translocation, the problem gene, has the regulatory region of skin collagen and the transcription unit of PDGF. And now she's going to express PDGF in the skin cell because the skin collagen will be activated. That same woman has the opposite gene present also, right? She's got the PDGF regulatory region connected to the skin collagen transcription unit. And that's not going to get made. You know, it's just goofy, weird, and useless. Yeah. Yep. Yeah. Okay, that's them all. And then Catherine.
We talked about how you can use the Greek fluorescent protein to look at very like specific things within organisms, like where myogenin is being.
Yeah, we can use that GFP strategy to create these, what could be called a reporter gene, because they're going to report the activity of MyoD, or report the activity of the myogenin enhancer, or report a muscle differentiation, and when myogenin gets expressed, right? So it's just kind of 1,001 uses for that strategy of making a transgenic organism like that. And that's just one example of a transgenic organism. There are a variety of different things that will come up in our course where we'll be like, oh, you can make this kind of transgene and make another type of transgenic organism. Yep. Yes.
Question about that. So in our neurobiology class, Ellie and I are kind of being taught about how GFP can be used to map brain connectivity, like neuron connectivity. Is that sort of like the same way that GFP is used in the
Yeah.
brain?
Yeah, here's an example of this. The professor of neurobiology, Doug Curry, and I overlapped in Seattle, Washington, so he and I knew each other back when I was in Seattle. And there was another student there, Kate. And Kate went on to study these zebrafish. And zebrafish are vertebrates, and so they're pretty closely related to us. What's valuable about them as a model organism is that they're vertebrates and they have lots and lots of orthologs, and they do a lot of similar things, like a lot of their development is really similar. For example, they have a face, which sounds weird, but vertebrates have faces. And their faces are made by cells called neural crest cells. And what's interesting and valuable about zebrafish is the embryos are clear and they develop outside of the animal. Right, so the mom lays eggs and dad releases sperm, and they have development outside the organism. It's clear, so you make a video of that. And Kate has her research starts with this transgenic zebrafish that has nerves that express GFP and glow, and as they go growing into the developing phase, she can see that happen, and that's her starting point. It's a regular, normal zebrafish with glowing nerves going into its face. And what she does is she screens them for mutations that have defects in face development, and she can see it because the glowing nerves are growing wrong, like that. So she can use that as a sensitive way of seeing what's happening in the growth of their face. And what she's studying are things like cleft lip or cleft palate, because that's development of our face, and our face gets put together. And zebrafish have that same kind of development, because other vertebrates do as well. And so, yeah, GFP can be used to make a transgene, get it expressed in nerves, and look at the patterns of nerve growth and nerve connectivity like that. Yep. Yeah, and one more is Joe. What's one other thing we talked about?
We were talking about the oncogenic retroviruses and how they can cause chromosome translocation. I kind of Isn't that like each oncogenic retrovirus causes chromosomal translocation, or is it only like MYC causes chromosomal translocation, or does each one?
That's a good question to clarify. So the oncogenic retroviruses do not cause chromosomal translocation. What happened is that MYC got discovered to be an oncogene, a cancer-causing gene, twice. Once in the 1970s as part of a oncogenic retrovirus, and it was because it just got expressed too much. Then second, completely different, in a chromosomal translocation, where in the blood cells, MYC got recombined through a chromosomal translocation. So two different ways that MYC got discovered to be an oncogene. One, in the 70s, in that, foot in the door, oh wow, look at these, few genes that must be oncogenes. Then second, in the 1980s, in a real cancer called Burkitt's lymphoma. I think it's a non-Hodgkin lymphoma, like you kind of have heard of that. And that's caused by a chromosomal translocation. So first thing is to clarify that. Two different stories about MYC, like that. So oncogenic retrovirus is not chromosomal translocation.
So it was discovered in chromosomal translocation, but it did not specifically cause it, is what you're saying?
It was discovered twice. Once is an oncogenic retrovirus in the 70s where it just got picked up. And then secondly, it was discovered like the skin cancer story where you're like, there's some people that have a certain kind of lymphoma, and when we look at their cells under the microscope, we see a certain chromosomal translocation, and they found that, and they knew that, they called that a certain kind of lymphoma based on that diagnosis. And then later, when they could really sequence the DNA, they were like, oh my gosh, there's that MYC involved in a translocation. And we'll look at that in detail in chapter 18 when we talk about MYC some. But yeah, good to clarify that, right? And that's one thing we want to do with these concept chats is to kind of recall and repeat. That's really helpful. Yeah. Okay, so just hold on because I think we're done talking. Let me just say, just on the last 1 minute, so, GFP as a tool, that's kind of a chapter 4 concept, is use these things as tools. And so then a second trick is that I'll talk at the start next Tuesday about using reverse transcriptase as a trick tool like that, because that's one that is talked about in chapter 4. And I'll just talk about its application just like that. This is an example of that. And then it's actually on to what we want to talk about chapter 9. So definitely read chapter 9. Recognize that it's the clay model stuff, but it's a lot of stuff stacked onto it. So we need to talk through some of chapter 9 in class and carefully help guide you through that additional stuff like that. So you don't need to scramble to write this down. I just want to show you that we're almost going to talk directly and roll through chapter 9. So get that read, write down questions, and then we'll start going after chapter 17 after that, and that's those receptors. Awesome.